Hello, this is another video to introduce you to the work we do in the DISCOvERIE project. My name is Judith Farrés. I'm head of collaborative research at Anaxomics BioTech, a small-medium enterprise based in Barcelona that specializes in biomedical data analysis using bioinformatics and artificial intelligence-based tools. I co-lead Work Package 7 with Dr. Arno Lucas from Emergentech. Again, a small-medium enterprise. They are based in Vienna. They develop software and data analytic workflows for digital health. The project DISCOvERIE investigates the relationships between irritable bowel syndrome, IBS for short, and mental and non-mental comorbidities with the aim of getting better treatments and better diagnostic tools. You can get the overall introduction to the project in another video posted in this channel by Dr. Javier Santos, the coordinator of DISCOvERIE. And now I will use my avatar to help me introduce the Work Package 7 objectives and activities.
Hello, welcome to Anaxomics Workroom. I brought here in some diagrams to help me explain a little bit the work we do in Work Package 7 in DISCOvERIE. Work Package 7 is named Systems Biology and Integromics. It analyzes all the data together from the different parts of the project. Here we have a diagram of the project. We see that we have a work package that is started doing a clinical trial. So in fact, there are 2 clinical trials with IBS patients and patients at risk of IBS, collecting data and samples. We have another part that is studying the intestinal barrier function in humans and also in model systems. We have another that studies the central nervous system and stress axis function. Also the microbiome, again, with model systems and humans, and also another part studying genetics and epigenetic factors in the disease. We also have a part of e-health monitoring that collects data from wearables. So all these work packages, they will analyze their data, the experts in each work package, and they will have elaborated data sets that then will come to the Work Package 7, and we will analyze them together. One of the outputs of Work Package 7 is potential biomarkers that go to Work Package 8, where they develop kits for diagnosis. The elaborated data sets come to Work Package 7, and we will use different analytical methods to analyze this data, trying to maximize the results, taking advantage of the different types of analysis. We will analyze the data as it comes using data mining techniques to identify correlations and patterns that define different pathological backgrounds. But then we will also use systems biology that I depict here with this human form. The dots represent the human proteins, the non-human proteins. The edges represent the relationships between those proteins. So all that is known about the biology of the human being, and then most of this data can be mapped to protein effects, and then we can analyze this data on the context of this network. This, as you can understand, it's very complex network, so there are many potential relationships to analyze. So we make use of artificial intelligence to analyze that. We will build different patient models that is represented here for each of the pathological backgrounds. And this will help make the model interpretable. So we could evaluate the relationships between groups of proteins, and will identify patterns. That is what we are depicting here. So not all the connections are feasible. The model identifies which ones are best connected. So here I'm showing the diagram of representing the clinical trial, so to help explain how we interpret this data. We will have IBS patients. We will have IBS patients with comorbidities, with multimorbidities, also as mental pathologies. We will have anxiety and depression. And as somatic pathologies, we have chronic fatigue syndrome and fibromyalgia, and we also have a healthy group. So within this data set, so analyzing the data across, we will be able to identify things that are similar in different groups and things that are different. So maybe we can identify what are the mechanisms that link or are different in the different types of pathologies or pathological backgrounds. We will also be able to identify key points that could help distinguish one from the other. So these key points could be finally diagnostic biomarkers that help identify which patients, for instance, let's imagine that we have one patient that it's IBS comorbid, but it's not known. Maybe these biomarkers can help identify that this patient is suffering also from other comorbidities. Then we have also another data set, a very interesting one, where some of these patients are followed along the time. Then, for instance, in IBS, we might have some patients that at the beginning, they all have IBS, but some will stay IBS all the time, but some others may evolve to a comorbid state or multimorbid state. So this data set is going to be very interesting because if we can identify differences at baseline for these patients, this could help us identify potential biomarkers that can be prognostic biomarkers, so that we could check patients at that IBS and see which ones are at risk of developing comorbidities and try to follow them more carefully, or even try to prevent this comorbidity from appearing. So, so this will be the outputs: mechanistics, understanding of the diseases, biomarkers, diagnostic and prognostic. And also, we will screen treatments to see which existing drugs will be screened to see which ones could benefit the patients the most. And some of these biomarkers are going to be developed into test kits. So with that, I finish the Work Package 7 description. I hope to come back soon with wonderful news from this project. Thank you very much.